May I introduce Dr. Vibhaskar, who leads the Scientific Advisory Committee at Pentacrate and also heads the Department of Genetic and Molecular Medicine at Kogilaben Birubai Ampanya Hospital, Mumbai. Hello, Dr. Vibhaskar. Thank you for your time on this podcast. It is a pleasure to have this conversation with you. For the sake of those who are not familiar with the zebrafish, could you highlight how you created the zebrafish model?
See, nice, kind introduction, Dr. Surya. See, Pentagrid has already established a CML model in Zebrafish. What we need to do is to transfer a cell line called K562. So that is a CML cell line into a bone marrow of Zebrafish. And after 6 days of transplant, the animal will display the CML pathology and phenotype with high WBC count, enlarged spleen. And when you do the test, you find it is positive. Positive for P210, 210 transcript, 210 transcript. So this most common BCR-ABL protein produced in CML patient based on loci of the chromosomal breaks. And they are P10, P190, and P230. So what we are going to talk about the protein that is P210, which occurs mostly more than 90% of CML patient. Am I correct? So other phenotypes changes that we see in the feces are fatigue, weight loss, reduced locomotion. Also, the CML model also display behavioral fever, and sometimes it is very difficult for the reduction of fever. It will be like a reluctant or rather aversion to reduction in temperature. So which leads to low survival rates at less than 30 days. This is what the observations have been made. So now in this model, the tyrosine kinase is mutated just as in human CML, what we see in human chronic myeloid leukemia, which increasingly phosphorylates protein involved in cell division. As a result, it leads to proliferations of WBC leading to CML. This is how you replicate a human CML into fish CML model. Over to you.
Thank you, sir. How do we mitigate the tyrosine kinase resistance, and what would be the future for Discovery Research Company?
See, Dr. Surya, before I enter into tyrosine kinase inhibitor or resistance, I'll give a brief account on how this chronic myeloid leukemia is a reciprocal translocations. It's a reciprocal translocation disorder rather, driven by breakpoints, cluster region, that is BCR, and Abelson leukemia virus, that is ABL, fusion genes, that stimulates abnormal tyrosine kinase activity. Right? So tyrosine kinase inhibitor are effective in treating PS chromosome positive CML patient. However, the appearance of TKI resistant CML cells is a hurdle in CML treatment. Therefore, it is necessary to identify a novel alternative treatment modalities targeting tyrosine kinases. Right? So, from studying gene sequences of resistant patients, this is what we know. There are conserved regions, right, of the kinase domain that are frequent sites of mutations. For example, the P loop. So from patient data, what we know that those patients treated with imatinib earlier are less prone to resistance. Hence, there is always a classical, opportunity to try to, um, various dose combinations and overcome TK resistance.
Your thoughts on alternative treatments modalities such as both BCR-ABL1 targeted and non-BCR-ABL1 targeted therapies. Say, for example, emacitidine and imatinib co-administration.
There are two ways of approaching new drug discovery in chronic myeloid leukemia. One is to identify the TKI, that is tyrosine kinase inhibitor, with a better drug response. That is better half-life, new target site, co-treatment with other TKIs such as dasatinib and tolontinib. The other is to use non-BCR-ABL dependent therapies where, for example, new drugs such as inhibitors of farnesyl transferase. So this is a good example. I'll give a good example of tinifarnib from Johnson & Johnson, which down-regulates RAS activation. So another promising non-BCR-ABL modality could be the target CXCR2, mTOR, and CML cascade. So this CXCR2 antagonist, what it does, it suppresses the proliferations of CML cells where cell cycle arrest in the G2M phase, and CXCR2 inhibition also weaken the mTOR and CMYC and BCR-ABL expression, leading to CML cell apoptosis. So here we use interleukin, that is IL-8, as a prognostic marker with CXCR2 as a drug target. So this could be a successful approach. And we have two modalities at hand that can be applied together.
What are your experience with patients of chronic myeloid leukemia, and what could be the future for them?
My experience is very interesting. I have seen more than 1,000 such cases. During my association with Apollo Hospital way back in 1999 to 2012, 11. And then now where I'm working now, Kokilaben Dhirubhai Ambani Hospital. So mostly we are engaged in diagnostic work. So we produce the best quality report for the management of patient, right? By the clinicians or the physicians who refer the cases. So at diagnosis, what my observation is, about 5% of CML patients, they lack PH chromosome. So this is the place where we need to observe more in detail using various, possible techniques or technology. Despite the presence of the BCR-ABL rearrangements, you find 5% of CML where pH is negative. So two mechanisms have been proposed about the occurrence of these rearrangements. First one is a cryptic insertion between chromosome 9 and 22. Okay? And the second one involves two sequential translocation, a classic 922 translocation followed by reverse translocation, right, which constitutes normal morphology of the partner chromosomes, right? Here, about 5 to 10% of newly diagnosed CML cases, those who lack PH chromosome by conventional cytogenetic, that is karyotyping, it doesn't mean that the involvement is not there, right? But they are shown to carry BCR-ABL fusion, which can be detected by other methods like RT-PCR or FIS. We have many cases where we have seen variant, BCR-ABL fusion to be positive. So these minorities of pH negative cases may harbor a complex cytogenetic variant involving not only third chromosomes, but there are reports where fourth chromosomes is also associated to be are involved. Besides 922. So this is and pH chromosome is very interesting. It is 90% found in CML, and 5% or less than 5% are seen in children. That is pediatric group in ALL, acute lymphoblastic leukemia. And you will find 20% to be associated with adult ALL. Okay. Whereas very less percentage, even 2% or even less than 2%, we find its association, the PS chromosome positive association with acute myeloid leukemia. And another interesting part is variant, as I described just now. So when you see such variant kind, so they are actually, we have observed 4 different patterns of rearrangements suggesting a high genetic heterogeneity. So generally what happens, we find, which is, the most common, we find 1 red, 1 green, 2 fusion. When we do FIS, it is about a, few number of loci. It is a gene of your interest. So it can pinpoint the gene that is present on 9 chromosome and then gene that is present on 22 chromosome, ABL and BCL, right? Now what you see 1 red, 1 green, 2 fusion. This is the routine, mostly say. But apart from that, where you find what? The location of BCR-ABL fusion gene, the derivative is 9. Also the derivative is 22 because 22 goes to 9, 9 goes to 22, right? But apart from that, you find 2 red, 1 green, 1 fusion, where the derivative is 22. And the mutation you find, the transcript, the molecular transcript and the wild type, you find the mutations, right? Now, in this, what you find, your derivative to be 22, where you find 2 red, 1 green, 1 fusion. Another variant, what we see while doing FIS, that is what I have noted down just to explain to you. 1 red, 1 green, 1 fusion. Here, the derivative is 9. Okay. Now, the molecular transcript is B3BA2, and it is wild type, the mutation, right? Now, when you see another, the 4th type is 2 red, 2 green. No, sorry, 1 red, 2 green, 1 fusion. So here the derivative is 9. So this observation of 4 different patterns of rearrangement, what it suggests? It suggests high genetic heterogeneity. So this not only can be done only by FIS. When you find such variant cases, you need to apply or employ different methods. Not only just a karyotype you did not find. Anyway, it did FIS. You did not able to pick up because other 3rd chromosome is involved or 4th chromosome is involved. So what you need to do is to do the karyotype and then look for that particular probe if it is available with you. If it is you are suspecting to be associated or to 3rd chromosome is involved on 4 or 7 or 10, wherever it is. If you have any probe available, or you have to go for the whole chromosome painting. So these are different, technology are available to pinpoint the diagnosis. So FIS is definitely very efficient tool. Apart from that, you have RT-PCR and DD-PCR, which are very helpful for minimal residual disease, to know. So that will be more effective rather than doing just, karyotyping when patient comes after 3 months or 6 months after the, medication is prescribed or medicine has been, started by the patient. So apart from that, you have high-end NGS-based genomics. Let me tell you one thing. See, karyotype cannot detect variants smaller than this. Okay. What do you mean by variant? It may be deletion, it may be duplication, it may be translocation, some rearrangements, structural rearrangements. Right? When you talk about FIS, what it can do, it has got some lesser number of loci. So it can target the gene of your interest. When you talk about chromosomal microarray, there is a little advanced technology. So there, what will happen? It can tell about more than 50 kb. So these three techniques actually in our routine day-to-day clinical practice that we do in our genetic and molecular medicine lab, these are the forefront of standard of care kind of tests that are being recommended. Okay. Now, although FIS analysis is efficient, RT-PCR is efficient, new technology have evolved. So next generation has come, and you will be surprised that there is another new technology now has been developed. This is called optical genome mapping. And this is otherwise called as the next generation cytogenetics or cytogenomics. Comprehensively, what it does, it identifies all classes of structural variants, copy number variations, repeat expansions, contractions, ring chromosomes, and absence of heterozygosity, aneuploidy, and I think triploidy. Sometimes you miss even in chromosomal microarray. If you are not using high resolution platform, that is Affymetrix. I have come across in my, practice. I have seen many product of conception women when they go through recurrent miscarriages, and suppose we use Agilent platform, whatever reasons it is done, not Affymetrix, so you may miss triploidy. Triploidy is lethal. It is associated with repeated pregnancy loss. I'm just telling the limitations, advantages, and disadvantages of different technology. Nothing else.
Could you highlight the role of zebrafish in new drug discovery for CML approaches?
Zebrafish's role in CML drug discovery is to solve, why we need to do, is to solve the problem of patient's heterogeneity. For example, we have over 60 variants which already available in Pentagrid of P-loop mutant zebrafish that resist imatinib therapy. You can always screen them if your new drug or combinations can rescue a population with a multivariant CML. You could profile bone marrow and spleen for hematology counts and measure disease course slowdown or reversal. So, Dr. Surya, I feel, this is probably the most difficult question, to address in the drug discovery scenario. Now, most of the times we hear like, will my fantastic and absolutely, a fantastic preclinical drug, will it work in clinical trial? Right? The answer is most likely. Why not? Since here you obtain ED50s, the median effective dose which refers to the dose of a drug or biologic, right? So from our 60 variant of diseased piece, then a single disease model. And this makes the chance of drug more likely to be successful in clinical trials.
Thank you, sir. Thank you for your valuable time.
Thank you, Dr. Surya.